So thanks. So I'll just go through and talk on the a very important topic that is typically on the disease management and sustainable farming. So as we all know that disease is the outcome of interaction between pathogen, host, and environment. So I will be taking the inputs and the information what we had generated here in ACR, specifically focused on the pathogen. So how the information linked with pathogen can help to devise the strategies for the disease management. And as we know that the mega trends in agriculture that is driving the modern agriculture, we see a lot of challenges there. And one of the big cause of those challenges is the population. And what we are seeing by 2050, we are going to add more than 2 billion population again, and that will require more than 50% food that is being produced now. So this is also aggravated because of a lot of loss is being happening, what is being produced because of the climate change, and also the arable land is decreasing day by because of the urbanization and all those. So it is putting a lot of pressure on the agro ecosystem, and that's where we need to use the natural resources very efficiently and responsibly. Over and above, the increasing pressure of the diseases and insects and weeds because of this climate change is also becoming enormous. And that's where the innovation in agriculture is going to really take this forward, where we need to really grow more on the small available arable land. And this is one of the example. If you see, by in 1940, to produce 250 kilogram of yield, we were yield of corn, so it was required to have 1347 square meter area. But with the innovation in the agriculture and a lot of technologies, as we were hearing in the morning from Dr. Channa Prakash also, so that much amount of this yield we are able to get now in 167 square meter area, if you see in this screen in 2019. Uh, and in future, with the advances in the technologies like the genomics, genome editing, and all those, so this area we can further reduce. Like in 120 meter square area, we can produce the amount of corn, for example, that was produced in 1940s in 1347 million square meter area. So that is possible because of the innovation. But the important topic here today is on the crop health, and we see that a lot of diseases that is becoming and appearing every year. They are posing a lot of risk on the humanity because crop is being lost. And some example here in banana, fusarium built, then in blast recently that was got introduced in Asia, for example, in Bangladesh. And last year we had seen in 2022 a new disease that was appeared in rice, specifically in North India. That was a viral disease for the first time. So these are the diseases that keep on coming and devastating a lot of crop, and that's where it becomes very important to address these diseases. But what is important when we talk on the disease? So as I told in the beginning, so three major components: the host, pathogen, and environment. We need to understand, and pathogen becomes more important because they keep on changing with the kind of host genetics and the change in climate conditions. So that is very important. And what we see, whatever resistance we are deploying as of now, they are not going to be the important one going forward because they are going to be broken. So in that scenario, the basic understand for the sustainable agriculture in terms of the disease management is whether we can monitor the pathogen shift very early and correctly, so that we can help the farming community to address the diseases in very accurately manner, so that they can save the yield loss. And second is also the strengthening the plant immune system. So we'll just go and take some example from rice, because rice is major crop in Asia, and that is a kind of crop that is helping humanity in terms of providing the sustainable food system for almost half of the world population. So and from plant health perspective in rice, so you see the diseases, insects, and weeds, they cause a lot of damage on the crop. And if we focus on the major diseases here, so you will see the bacterial leaf blight, that is important one, rice blast and brown flower trapper insect, specifically in this region. So we'll take the example of bacterial blight disease, and what we have estimated around 30 million acre area is affected annually in the region, and it causes a lot of damage to the crop. From looking from the topic, specifically from the pathogen side, so we will go more deeper, but if you see the disease per se, so it can cause, means it can come at the flowering stage and it can destroy the crop, and there will not be much to harvest. So it is important because the pathogen of this disease, that is bacteria Xanthomonas oryzae, Pathobar oryzae, is having a lot of variability. And if you see on this slide, so we could identify 12 major pathotype out of 1410 isolate that we have characterized as of now. And none of the single genetics, what we see here, XA1 to XA23, is able to protect at all. So that's so that pathogen variation is very important. And that's why its monitoring becomes very important to save the crop. And also, if you see the distribution patterns, so they are widely distributed. Like, for example, all these races, 12 races, so they have very wide distribution across all the regions in India. Some of them, they are having high population, like third pathotype is having around 38, 39% population structure, but still there are others that is present in all other geography. And the kind of risk what is seen, like as of now, some of the major genetics that has been deployed. So when we search that what is the spectrum of their effectiveness and what are the percentage of pathogen isolate that can break it, so we found that XA21, the major gene that is being deployed by public and private sector, so there are already around 14% pathogen that can break it. So the major question is, if we can really detect them it quite early in the cropping stage, then that becomes easy to manage them in the field itself. So how we want to advance those technologies to detect the pathogen very accurately and diagnose. So as of now, it is more on the manual, like sample collection and then doing a screening in control environment, or in controlled field condition. But that is not a kind of high throughput. So we need to go ahead from manual to more automated and predictive way, utilizing the genomic knowledge and the genomic understanding. And that is the major shift we are talking about in the pathogen understanding. And that's where we started collaborating with different institutes across globe, like for example, in India, CCMB and IMTech. So we just went ahead and did the genome sequencing of 100 isolate to understand that how their genomic architecture looks like. And based on those genomic architecture, we were able to pinpoint what are the major changes in the genome structure that define and decide new race development. So if you see on this slide, so based on those genomic architecture, we were able to identify the major change in the pathogen genome, which is making that particular race to cause disease on XA21, for example. So it was like any isolate, if they are liking this RAXX gene or they are mutated at that locus, so they are able to break the XA21 resistant gene that is deployed. So the whole point is that if we are able to identify these and we are able to pinpoint these regions in the genome, so we are also able to make some assay so that we can start detecting that kind of race directly from the field sample. And that is the direction what we are taking. Not only to that resistant gene, we have also tried to look some of other major resistant gene in bacterial blight like XA5 and XA13, because now we are deploying the genes in combination also. So these three resistant genes like XA5, 13, and 21, they are the major ones. And we were able to identify the genomic region for the effectors in the pathogen, which are helping them to really break these resistance genetics. So, like for example, if you see these two effectors like PTHX1 and XO4, XO7, if they are present, then these isolates, they are the virulent on the XA5, means they can break XA5. And same way, if PTHXO2 and 3, if they are present in any pathogen isolate, they can break XA13. So that's where we are taking all these examples into consideration to devise some of the technologies going forward so that we can start detecting the pathogen directly from the rice tissues, for example, in the field. And now, since the genomic technologies, they are very advanced, and we see that it is possible in the field itself, like some of these sequencers, nanopore sequencing system that is now available. We can take that machine, it's very small like our phone, and we can take this in the field, and we can just take the sample and crush it, and then we can just do that the sequencing itself. So that way it becomes very efficient and also important. We can avoid a lot of process during the pathogen isolation and screening. So we can just go in field and see that if any field is affected, so what is the race, whether it can break the genetics that we are deploying or not. So that way it becomes very easy. So this is the approach that we are following, and that we are going ahead to monitor the pathogen quite early in the field. And it is not limited to the one disease. Like this is the disease that we have made a lot of progress, but we are also trying to address with other diseases also fast. So rice blast, we have seen that it is having a lot of variation in the pathogen. So that's why we need to understand this also. So similar approach we are taking in this one also. Even in other crops like in corn, so like for example stock rot complex, so that is also a major disease in the region where multiple pathogen is involved. So it is really important to understand what is the pathogen that is able to cause the disease, and how accurately you are able to diagnose and recommend the kind of important genetics that can be deployed in those geography that would be effective against the dominant fungus, pathogen, or species or race. Same way, we are working on another disease in corn, that is downy mildew, and that is important specifically in Philippines and Indonesia. And that disease is caused by three species of Sclerospora, that is Medes philippiensis and Sorghai. But we need to really understand what is the dominance of these species in different regions in Asia, so that the genetics that we are going to deploy, it should be correctly deployed in those important regions so that it can provide the required level of tolerance. Otherwise, if genetics is resistant to one species and susceptible to other, then it will not be effective and cannot save the crop. So that is how we are looking, and that is how the whole scientific community is trying to address the pathogen risk, means detecting them in proper time, predicting the pathogen race evolution, and also going forward modeling that the risk. So that is the way forward. But that information is also utilized to strengthen the plant immune system, like the kind of varieties and the products, hybrids they are being produced. So they are having good level of tolerance or resistance against those evolving pathogens. And the example what I talked on the pathogen. So based on the pathogen information, the resistant genetics that we are trying and we have deployed, so that is working well. For example, here you will see based on all these pathogen information, we looked at what are the genetics that is effective against all those 14, 1500 isolate. And you will see here in green color that is showing the effective genetics like it's 13, it's 23, 21. So some of those genetics has been deployed and they are in the region, and you can see the kind of effect they are providing. If resistance is there, so they are green. If there is no resistance, then they are destroyed by the disease. And you can see the yield loss, for example, if there is no resistance, 1.7 ton, and if we have resistance, 8.3 ton. So that way, it is helping a lot, and we are putting the right genetics in right geography based on the pathogen understanding. And that's where it is making all those genetics more sustainable and providing a lot of relief to farming community so that they can do the the farming in more sustainable way by managing the disease. One example is the way it is helping the farming community. I will take this example of one trait, the bacterial blight. So we worked on and we launched first product in 2008. I'm just talking about the product that Bayer is putting in the market, means that is being grown by the farmer. So that product only is being grown in now around 4 million acre area and saving a lot of means the loss for the farmers. And also, as we all know that for bacterial and viral diseases, there is really very difficult. They are very difficult to control. We don't have a good chemistry. And for example, in bacterial case, a lot of antibiotics are used to manage, but that is not good option because that antibiotics they go into the food chain system, and we as human being and our animals they are becoming resistant to antibiotics as well. So that is not good practice. So that way they are really helping a lot, and they are means farmers they are saving a lot on growing these resistant cultivars. So this is not only the Bayer or other private companies, a lot of public sector institutes they are also coming with a lot of varieties that is resistant to these diseases, like for example NRI, then IIRR, IRI, PAU. So all those institutes and universities they are bringing a lot of product in the farming system. So that's where the farming is going to become more sustainable from the disease perspective. And 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 what impact these products that they have brought in the farming community. So you can see here. So we have launched first product in 2008 as Rajdhani BLB resistance, and then we brought BLB plus BPH resistance in 2017. And one example here, the farmer is being recognized by our honorable Prime Minister by growing one of the our product, and there that farmer was able to realize enormous yield, like 11 quintal yield per acre in the variety versus 30 quintal in the product that was bacterial blight resistant. So that kind of impact it is bringing in whole farming community, and also it is making the farming more sustainable. And this is the journey, for example, in Bayer itself. So we started putting the product in 2008, and now we have multiple products with multiple stress tolerance, and that is helping the farming community a lot to manage their crop. And going forward, our innovation will also help to define and design some tailored solution where we can integrate multiple options together based on the disease prediction. We can recommend that this variety, this chemistry can go together so that farmer can really have a tailored pest management solution going forward. So that is the direction that we are taking going forward, and that is going to help our sustainability commitment that we have made in United Nations, that we are going to help to reduce the 30% greenhouse gas emission by 2030, And we are also reducing the environmental impact of crop protection chemistries by 30%, the way we are developing the resistant products, for example. And we are also empowering the smallholder farmers across the regions. Our target is to reach 100 million smallholder farmer by 2030. So this is our commitment to smallholder farming community, and the way we are working, and the way we are advancing the innovation and technology. So we are very sure that we are going to touch the farming community and help them to grow healthier crop going forward. So all these activity was done in collaboration with different stakeholders. For example, CCMB, these scientists, Dr. Ramesh Sonti and group, then IMTech in Chandigarh, Dr. Prabhu Patil and his group, and then IIRR, Dr. Ramesh Sundaram and his team was there. We collaborated, and ERI, International Rice Research Institute, Dr. Mira Cruz, and also in California University, Dr. Pamela Ronald, Colorado State University by Dr. John Leach and group. So this is the continuing effort, and that we are on the right path to define and design the technologies that can really help to farming community in better way. And I believe we all together, whole scientific community is moving towards that direction only to help farmers to grow healthy crop and have less crop damage. So that's all from my side. And thanks a lot for this opportunity. If there are some questions, I will be happy to take. Thank you.
Ji. Thank you, sir. Is there any questions for him? Ji.
Good morning, a great lecture. Hello?
Good morning.
Right.
Yeah.
So I just want to ask if the sweet proteins have been now been advocated to be, able to protect plants from young summers infection. Do you have any
Hello.
comments on this? Should we work on sweet proteins? Sweet proteins also reduce plant growth also overexpression. Right.
Yeah. So yes. So if I could understood your question, so you are talking about sweet protein. So means there are a lot of effort that is being done on that direction. And when we were working with ERI, so they were targeting some of those genetics to edit, because those are the sweet genes, those are targeted by the pathogen. So what we have seen, those genome edited lines, they are not having any yield drag as of now. So as Dr. Channa Prakash was also mentioning in the morning, so whenever we are doing or we are editing some of those genetics, we need to be really very careful, and that need to go in enormous testing, so that we are making sure that there is not any kind of yield drag and all those. But what we did is through conventional breeding, and what we have found, there is no yield penalty and all. But there are certain reports, you are right, that sweet genes, because they are the sugar transporters, if they are being hijacked by the pathogen, maybe because those sugars, they are required for the grain filling also. So means we need to really test enormously before launching any product. But as of now, we haven't seen any yield drag.